So this presentation is going to be an introduction overview of light sheet microscopy, and then some application with regards to fluorescence fluctuation spectroscopy. So most of the microscope techniques can be classified in two categories, which are the camera-based approaches and the single point detection approaches. Of course, you can have combinations in between, just as the talk was alluding on before, with this small kind of, you could also call it a nano camera, I guess, a small spot array. Okay, so in general, for a simple camera-based, the most simple camera-based fluorescence microscope would be the epifluorescence microscope, in which you illuminate the sample in the entire axial direction. So everything in the field of view on, below and above is excited. Then you collect the fluorescence and you image it on your array detector, which, for example, can be a CMOS camera or similar, which has many, many pixels. And the advantage of this is that it's very fast. You can measure all the pixels in parallel, snap an image in a few milliseconds, and be really, really fast and get a large field of view. The disadvantage, if you implement it in this way, you get no optical sectioning. You excite molecules that are above and below the focal plane of your detection lens. And you also collect this fluorescence, which gives you kind of a diffuse background if you have a thicker sample. The other way is to do single point detection, in which you focus your excitation beam into a single spot in the sample, diffraction limited spot. And then you collect your fluorescence using a single point detector, such as a photomultiplier or an avalanche photodiode, for example. And with this concept, you can, for example, introduce a pinhole before the detector, which will block fluorescence light that is coming from out of focus planes. Or you can use nonlinear excitation, for example, 2-photon excitation, to limit your excitation volume. And in this way, you can get optical sectioning. However, to get a full image, you need to scan point by point the entire sample. And because of this sequential data acquisition, this laser scanning microscopy is typically much slower than camera-based approaches. So the light sheet microscope was trying to combine the best of both worlds, so to speak. Light sheet microscopy is often known as single or selective plane illumination microscopy, or SPIM. And here the idea is that you have two objectives. I mean, there are some other implementations that use a single objective and some tricks to generate a light sheet, but most of them have two objective lenses. And the idea is that you have one lens where you collect your fluorescence, and then you have another lens, typically at a 90-degree angle, that generates the illumination in the form of a sheet of light. And because this sheet of light only illuminates the plane that is in focus of the detection lens, you don't generate any out of focus fluorescence, and you can have optical sectioning, but with a camera detector. So some of the advantages, you have this optical sectioning capability that is similar to a confocal microscope. You can use cameras with many, many pixels, so you can have really a large field of view or volume if you do Z-stacking. You don't need any dichroic mirror to separate out the fluorescence light from the excitation light. Those CMOS cameras have very high temporal resolution, on the order of 10 milliseconds or 100 frames per second. And another big advantage of this technology is that because you're only illuminating the plane that you're actually imaging, you have much less phototoxicity compared to the confocal microscope. In the confocal microscope, while you're only detecting from the plane of interest, you're still illuminating everything that's above and below. So if you think about 3D data acquisition, you have much more photobleaching with the confocal microscope. Okay, I just want to give you a little bit of background, just one slide on the history of light sheet microscopy. So interestingly, this concept was already developed in 1903, so more than 100, almost 120 years ago now. Here you see a schematic of the initial drawing where they actually collected light from the sun and then used the lens to generate this perpendicular illumination. And then this was used to study colloids, I think gold colloids in solution dynamics, and eventually this led to a Nobel Prize in 1930 in chemistry, not on the microscope itself, but yeah, on this investigation of those colloids. But then somehow it was not used that much anymore, this kind of system, until 1993, where it came back with the acronym OPFOS, and then in 2004 under the acronym of SPIM, before they called it the ultra microscope. So one could ask, why is this? Why was there not so much interest in between? And the reason is that many other components that are now standard were missing before, like cameras. In the original implementation, you would have just looked with your eye. There were no CCD or CMOS cameras. For the light sources, there were no lasers. And then, of course, all the other electronics, computer data processing to collect and be able to store and analyze all this data, the huge amounts of data that this type of microscope can generate. So when this came up in the 90s and 2000s, then this technique really took off. And now we have a really huge variety, different implementations, applications of this kind of system. I'd just like to show this slide, but it's by no means a comprehensive list of all the systems that are available. Okay, so how do we get the light sheet? How can we generate the light sheets? Generally, two principles are utilized. And one, the more simple one, is to use a cylindrical lens. So a lens that focuses the light only in one dimension. And if we place this in the excitation path, yeah, it focuses it in one dimension, but not the other, generating this kind of sheet-like illumination. You can also see immediately one of the issues of light sheet microscopy, and there have been many inventions and publications on how to address this problem, which is that the thickness of the sheet is not uniform. So you focus the light, it becomes thinner, then it reaches its thinnest point where you place your detection lens, and then it diverges again. So at the edges of the field of view, your Z resolution is not as good as in the center. But this, people are trying to address this with other illumination schemes, like, for example, Bessel beam, Airy beam, that can sort of push this further. So the other way to generate a sheet of light is to use a scanning mirror. So you just use an objective lens to low numerical aperture, typically, to focus your light into a beam, which then forms a kind of line in front of the detection lens. And then what you do is, during the camera exposure cycle, you scan this beam across the field of view at least once. And thereby you generate this virtual sheet illumination. And the advantage of this is that you're flexible. You can change the illuminated region by changing the scanning amplitude. You can use, for example, multi-photon excitation because you're focusing all your light in the single line. So there are a couple of advantages of this way of generating a light sheet, but it involves a little bit more of effort because you need these galvo scanners here to scan behind the objective lens. The other question is, okay, so typically we have these two objective lenses. So how do we implement this mechanically? How do we put our sample? The light sheet instrument is also very popular for animal imaging, like for example, the zebrafish. So they would be placed in, for example, hydrogel or a cylinder in between the objectives here. But what can you do if you want to image cells on a surface, for example? Well, one popular implementation is the upright configuration, where you have those two objective lenses at the 90-degree angle, and then you dip into your sample dish from the top. So this is an example of how such an instrument could look like. So you see that most of the components are assembled on this optical breadboard, which is now brought up vertically. And then down here, you see the stage that holds the sample dish, and then we have the two lenses dipping into it. The bigger one is the lens for detection, high numerical aperture, and then we have the excitation lens to generate a sheet of light, which is typically of a little bit lower numerical aperture. So this in this implementation was the scanning approach. So we have some fiber coupled illumination sources that are then reflected onto this galvo mirror scanner. We have scan lens, tube lens, and then can generate the sheet illumination here. And then we have the camera on the left-hand side, and you can have, if you want to have multiple channels at the same time, you can, for example, buy or build your own splitter. So you can have two color or polarization or other channels side by side on the same camera chip. And for live cell imaging, of course, it would also be nice to have some capability to heat the sample to 37 degrees typically, and maybe add some CO2. So how do you acquire the 3D images now? Well, you saw we have this kind of, with respect to the sample plane, the 45-degree angle. And what you can do is you can just move the sample, for example, with the stage through the sheet at a constant velocity, acquire the images at the same time, and then with the step size, you can reconstruct the three-dimensional images. Alternatively, you could also move the detection lens, but then you also need to move the light sheet, and both need to synchronize to always stay in focus. So that scanning is a little bit more complicated to implement. So what are the speeds that we can expect? So many of the modern CMOS cameras can do 100 frames per second at full frame. If you crop a region, you can go even faster. So for example, in this case, we imaged 200 by 200 by 60 micron cubed in roughly 10 seconds, which if you compare it to the confocal microscope is of course much, much faster. So here is an example of the type of images that you can get. Okay, let me play the video. So this is a 3D data set, and what you see here are CHOK1 cells expressing EGFP paxillin. Paxillin is this molecule that plays a role in focal adhesions that you have already heard about in the Briggs and number and brightness lecture. And yeah, you can immediately see the fast image acquisition. We can follow the motion of the cells in 3D. And you also see that we have minimal photo bleaching, which are all the advantages of the light sheet instrument. So what kind of information can we get from these data sets? Well, what you can see clearly by eye is the cell migration. You saw these cells kind of crawling on the glass, and they are assembling and disassembling those adhesions as they move. And this motion is on the order of minutes to maybe even hours. Then we have the structural changes, which is the individual adhesions, assembling and disassembling, which is more on the second to minute time scale. But then we also have the biomolecular dynamics, as already discussed previously, which is the single penicillin EGFP molecules in this case, diffusing in the cell and moving to different locations, in this case, in the end, to those focal adhesions. So we can observe many different processes happening on different timescales with this kind of instrument. Now the question is, okay, from the image data, just by looking at it, we can study the cell migration and the adhesion dynamics, but we would not be able to tell how fast the molecules diffuse within those cells. So to get this information, we can turn to image mean square displacement analysis, for example. Just to reiterate the concept really fast. So what we do is we acquire a sequence of images very rapidly. In this case, we have camera images and everything is exposed at the same time. So the one image by itself does not contain any time structure, as opposed to a raster scanned image where we then can use rigs to leverage this time structure. But instead, we collect many, many frames very fast as a function of time. And then the time structure lies within this additional dimension, so the frame-to-frame correlation, which we were not considering in rigs, for example. So we calculate the spatial temporal correlation of this set of images, and then we end up with this correlation function stack. And what you see is a peak in the center, which represents the probability to find a molecule at a certain distance. And the reasoning here is that if you look at very short time delays, then the molecules were not able to move very far away from where they started. So this probability distribution is very narrow and the amplitude is high. As we increase the lag time, the molecules were able to move further and further away from their initial position. And as a result, this function decreases in amplitude, but also broadens because now you have a chance to find a molecule further away from where they started. So this is what you can see here at increasing lag time. We have this broadening of this function. And by just fitting a Gaussian model, we can extract the width of this function as a function of the lag time. And this turns out to be or correspond to the mean square displacement of the molecules. So we have the MSD plot we can get from this type of analysis. And just to show you a few examples, one is the diffusion of a dye, a small molecule, rhodamine 110, which diffuses at about 400 micrometers square per second, which we were able to confirm with this method. Then you have the green fluorescent protein, which is a bigger molecule. Yeah, it's a protein, which diffuses at about 90 micrometers square per second. And then down here, you see some beads, which are even bigger, which in this case diffused about 30 micrometers square per second. So you can see with this method, although your acquisition is in the millisecond, on the millisecond time scale, and your diffusion time considering the molecule moving from one pixel to another pixel is on the sub-millisecond timescale. We're still able to capture the diffusion because we are using the spatial information in combination with the temporal information to get this kind of information. Okay, so here are some examples of application of these techniques in live cells. On the left-hand column, we see a cell expressing the green fluorescent protein. The shape of the cell, you think, looks a little bit different than what you're used to. And this is because now you have to remember we are looking at it from a 45-degree angle. So we're kind of slicing it in the XZ direction. So this is why it looks maybe a little bit unfamiliar. So down here is where the cell attaches to the bottom of the dish. Okay, so in this region, we applied the image mean square displacement. And you can see we get about 22 micrometers square per second, which is what you also were getting with other methods like RIG, single point FCS, and so on. Then we have our EGFP pexillin that you have seen in the previous movie. And we are getting about 6 micrometers square per second of diffusion in the cytoplasm of the cell. You can also nicely see that this protein does not go into the nucleus. Then we have the glucocorticoid receptor labeled with a red fluorescent protein, which does go into the nucleus. And if you look at the diffusion inside the nucleus, we got about 1.6 micrometers square per second. These are all cases where the diffusion, at least what we observed, was more or less free, which is indicated by the linear relationship between the mean square displacement and the lag time. Of course, it might be that if you look at even smaller lag times, that on smaller spatial scales, there could still be some trapping, but at least in the long range motion, for those three, we did not, we saw pretty much free diffusion. But of course, you can have other scenarios. Here, we have an example on the left column where the lysosomes were labeled with a green fluorescent dye. And if we apply the IMSD analysis to this kind of data, you see that it's not a linear relationship, but you have this kind of parabola shape. So it goes with the square of the lag time. And this indicates active transport, which is true. Those lysosomes are actively transported inside the cell. And in this case, we got an average velocity of 0.3 micrometers per second. In the center, you can see clathrin fluorescently labeled with the M-cherry fluorescent protein. And here we analyzed 2 different regions. One is where there were no spots, so no clathrin pits, and we get more or less free diffusion again. But then you also have these clathrin pits that were internalized, and they are actively transported again. As a control, you can see here the H2B EGFP, which just binds to the DNA in the nucleus. Yeah, in this case, we don't get any motion at all, at least not on that time scale. Okay, another approach you can use to get some information about molecule dynamics is fluorescence polarization or fluorescence anisotropy imaging. Here the idea is you excite your sample with polarized light, and then you detect in 2 channels, which is the parallel and perpendicular direction of the polarization. And the idea here is that if the molecules, if your fluorophores are static, they cannot move, and you excite with polarized light, then the emission should also be polarized, at least to a certain degree. But if the molecules are able to rotate, and they rotate before they had the chance to emit the fluorescence photon, then the polarization, so because the orientation of the molecule has changed, the polarization of the emitted fluorescence will also change. So yeah, you see a decrease in the fluorescence polarization the faster the molecules are able to rotate. So this is a measurement of rotational diffusion. So this is illustrated here on the right-hand side. We see rhodamine 110 mixed with water on top of a PMMA polymer layer that also was infused with rhodamine 110. So in terms of intensity, there's not any difference because we adjusted, we matched the two concentrations. But then in the fluorescence anisotropy, you can clearly see the difference because in the water, the rhodamine molecules can rotate fast, but in the PMMA, they are stuck and cannot move. Yeah, you can apply this principle to cells. Here are some CHO cells on top expressing actin, EGFP, and on bottom EGFP paxillin again. And what you see here actually is that in general, the polarization is already pretty high, 0.3 and even higher. The reason being that this EGFP paxillin complex is already a pretty large molecule. So it's not able to rotate much before it emits the fluorescence photon. But then you see those adhesions, they appear in a different color code, which indicates lower anisotropy. Why is this? Well, here those molecules are highly compacted, so you can have energy transfer between two EGFP fluorophores in the form of homo-Fred. And this also leads to a depolarization of the fluorescence. And this is what you can see here. If we quantify the polarization as a function of the Z depth, you can see that it's low near the surface where you have those adhesion complexes. And because this is slide sheet microscopy, you can have these 3D data sets, acquire them very fast as a function of time. For example, to track the formation of those adhesion complexes. Okay, so this was done on this upright implementation of the microscope. It works nice, but we had some kind of limitations that we wanted to overcome. Once we want to use a small sample volume, not have this entire dish. It would be nice if we can isolate the optics and the sample for difficult specimen. If we could have access from the top, for example, connections to add some fluids or something. And the horizontal orientation of the image plane is also advantageous if you think about a sample that is a layer to maximize the field of view. And we also wanted a more quick and easy sample change. So for this, we implemented light sheet illumination on a conventional inverted microscope, which you can see here on the left. You see the X by Z stage, the detection lens from the bottom, and then all the components to generate the sheet of light were assembled on this optical breadboard on the side. So we have a single photon excitation. We also have a 2-photon beam, and then again we implemented using the Galvo scanner to generate the sheet of light. Here is a close-up view of the sample chamber. Because we now have to illuminate from the side, we designed this custom chamber. We have cover glass from the bottom and from the side, and then we have those imaging wells where you can place the sample on the right-hand side. You can see this type of chamber inside the instrument with the two lenses. Okay, now some considerations about the geometry. So here you see the sample, a drawing of the sample chamber with the detection and excitation lens. If it's filled with some medium, water-like medium, and you have your object of interest in the center, then there's no issue. You can just take your data and that's it. However, if your sample would be at the bottom, for example, the cell would adhere to the bottom of the chamber, then you would have a problem because now you have a mismatch of the refractive index between the glass that the excitation beam has to go through and the medium, which is on top. So in this case, the light sheet would no longer form correctly. So you have to take into consideration, if you want to image this way, with a properly forming light sheet, you need to match the refractive index. So you need to support your sample with some material that has the same index as the medium that you put on top. We also did some calculations on how large can the mismatch be before you get noticeable aberrations. And here you can see the cross-section of this beam that is used to generate the sheet of light. You see the intensity profile as a function of the refractive index change in percent. So we saw that the beam waves starts to diverge at about 0.3 or 0.4% of the mismatch in refractive index, and you can also see that the amplitude goes down a lot. So the main effects of the mismatch lead to a translation of the beam along the optical axis, a shift of the beam normal to the interface. Those are not a problem because you can correct just repositioning the light sheet, but then you also have effects that are broadening in width and decrease of the amplitude, which you cannot easily correct. So what we found is that you need to match your index better than 0.3%. Of course, it depends on the illumination numerical aperture. If it's less, then you need to be less precise. If it's more, you need to be even more precise. So this is 4.3 illumination and 1 millimeter imaging depth. Here are some materials just to give you an idea what the refractive indices are. Marked by the blue box is water, PBS buffer, and some cell medium, which are in the range of 1.33, 1.34. Then down here marked with the green box, we have some popular hydrogels, for example, collagen and agarose. At those concentrations, they have a refractive index pretty similar to that one of the water or the cell medium, so that should work very well, and people have used that a lot. But then there are also some polymers, so they come in liquid form. You can pour them and then you UV cure them to polymerize, and you can also get some that have a refractive index that is pretty close to water. Just here to recap with this polymers, here are some beads in agarose. Of course, you have no problem imaging them with the resolution, in this case being about 370 micron in XY and 1.1 micron in Z. And here we have those same beads, but on top of this resin that has a similar index. And you can see that this approach also works well. Unfortunately, the resin has a little bit of autofluorescence, as you can see here in this XYZ view. Okay, so one example how you can use this is shown here. So we deposited this polymer, and then we deposited giant unilamellar vesicles with UVs that were anchored to a BSA biotin coating of the surface. The vesicles were labeled with the dye Texas red, and we had two different lipid compositions. One is just one lipid DOPC, which is fluid, and then a mixture of DOPC, DPPC, and cholesterol, where then you have phase separation, which form these kinds of domains. And those are not static, of course, but they can move, diffuse on the surface of the GUV. They have different affinity for the dyes, so that's why you can see them. And now the advantage is we can acquire 3D data very rapidly, in this case at about 1 3D data set per second. And then we could, for example, track the motion of these domains on the GUV surface. Here is an application in cells. We have lysosomes labeled with the lysotracker. Again, 3D data sets were acquired. And I just want to point out that this data set encompasses 500 Z-stacks. And if you would try to do that on a confocal microscope, you would probably already have bleached the sample before you can even get to that number. And then you can also use this to calculate the tracks and the velocity of the lysosomes in this case. Another example here is the zebrafish embryo. This was a 36 hours post-fertilization embryo labeled with the dye in red. And what we imaged here was the microcirculation of the red blood cells in the circulatory system of the zebrafish. And here we acquired Z-stacks at a rate of 5 stacks per second. So now you're approaching video rate imaging, but in 3D, not just in a single plane, but in all 3 dimensions. Okay, another application with regards to correlation spectroscopy. If you remember, the 2D pair correlation function method has already been introduced, and here the caveat is that you need optical section data and you need to be able to acquire it very fast, those conditions are fulfilled, for example, by the total internal reflection microscope. But in this case, you could only look at the membrane of molecules. But now, of course, with the spin providing this high spatial temporal resolution, you can apply the 2D PCF approach. So the idea was just to also quickly recap. We have a pixel of interest that we correlate with other pixel, with surrounding pixel at a certain distance. We call the pair correlation distance. And we repeat this operation for all pixels within this time stack of images. Without an obstacle, all of these separate correlation functions should look similar. Because there's no reason as the molecule, the motion of the molecule is the same in all directions in the case of isotropic diffusion. Now, if the molecule or the molecules would be near a barrier or an obstacle, the correlation on the other side of the obstacle would be absent or delayed if the molecule can move around. And this asymmetry or anisotropy in the direction of the correlation, we can quantify by determining the long and short axis of this correlation function. And then we can visualize the barriers to motion of molecules in the image. So for example, here, if you would have particles moving in a box and you would have a thin channel in the center that you cannot see, but once you apply the pair correlation principle, you would generate this anisotropy map, and you would see that the motion inside the channel is highly directional. And you also see those 2 weaker lines on the outside of the channel, which are because the molecules on the bottom left and top right, as they bump into the channel, they cannot cross, and we also get an asymmetry in the correlation function. So here's an experimental example on the light sheet microscope. So we have cells expressing EGFP suspended in the collagen matrix, and use the light sheet to generate optically sectioned data in one plane very rapidly. On the bottom, you can see the intensity distribution where you cannot see many features. But then if we look at the anisotropy or eccentricity, we can see all these barriers that exist to the motion or diffusion, in this case, hinder the diffusion of the EGFP in the cell. You can also do this in tissues. Here's an example of, this is actually the root of a plant, Arabidopsis, which is also expressing the green fluorescent protein. Again, in the intensity image, you cannot see much. You can maybe see the nucleus of the cell. But then if we turn to the anisotropy map, or the map which characterizes the center of mass shift this pair correlation function, you can again clearly see the barriers that exist to the motion of the EGFP molecule. So to summarize, the light sheet microscopy in general is very nice because it can accept a wide range of sample geometries, as you have seen, from single cells to UVs, organisms such as zebrafish, tissues. You can image all these. You can apply fluorescence fluctuation-based methods such as IMSD or the 2D pair correlation function. You can get 3D data sets in a very rapid manner. And also, of course, you can have multiple channels, as you have seen with the polarization imaging, for example. Okay, in the last few minutes, I just want to show you. You could maybe call it a toy, but we are thinking about concepts, how to use it for in-field research or some experiments. So here the idea was, how small can you make a light sheet microscope? What are the minimum hardware requirements? Well, we need some kind of light source for illumination. Then we need a method to generate the light sheet, which is, in the simplest case, the cylinder lens. And then we need a lens for imaging. And if we want to use fluorescence, we need a filter to select for that. And of course, we need a camera. But as you know, all of your mobile devices, your smartphone already has a camera. So could you actually implement a light sheet microscope using that? So this is what we came up with to try. So just on some plate that we had lying around, we mounted a small laser diode and the cylindrical lens, as you can see, and then some sample holder. In this case, we just had a small capillary with square faces, which contained the sample. And then here on the right-hand side, you can see the lens with the color filter for fluorescence detection, perpendicular to the excitation. And this interfaces with the camera of a phone in this case. On the most right-hand side, you can see a picture of this sample cuvette, which is a small capillary 1 millimeter cross-section that we were using to contain the sample. So what can we do with this instrument? Here are some images of 1 micron beads. This is just scattered light. So what you can see on the left is the cuvette or the capillary, which also scatters some of the light. And then you can see the beads inside. Because these are all cheap optics, you see you have some strong spherical aberration. But in the center of the field of view, it's actually quite usable. What you also see is, if you first look at it, you may think that the concentration of your beads is higher at the edges, because it looks like the density there is higher, but this is not the case. The reason why you see less beads in the center is because, if you remember, you have a certain light sheet divergence. So the thinnest point, the best Z section you get only in the center. And then in the edges, you illuminate more beads. That's why it looks like you have more, although the concentration is uniform. If we focus on this region where we have minimal thickness of the light sheets, we can take the cross-section of the beads to characterize the resolution of this instrument, which was about 3 micron with the optics that we used. Okay, so what can we do with this? Well, we can again measure the diffusion, for example, here where fluorescent, yellow-green fluorescent microparticles of 100, 200, and 500 nanometer, and we applied the image mean square displacement method to the image data. And as you can see, we can get the diffusion coefficients of those microparticles in water, as we would expect. So what about some other samples? So what we also tried was just to put a zebrafish which was actually unlabeled in this capillary, and then imaged it, excited with 440 nanometer excited autofluorescence of the fish. Okay, but then in this case, you would only be able to image one plane. So what about 3D image data? Well, we just were exploiting that because the fish was suspended in glycerol. If you flip this instrument upside down, which you can do because it's very small, the fish will sink to the bottom at a mostly constant speed. And while taking these images, we also were able to acquire 3D data. And on the right-hand side, you can see the reconstruction of the zebrafish autofluorescence. Another advantage of the light sheet again is if you stay in one plane, you can acquire image sequences very fast. So in this case, we were focusing on the heart, on the beating heart of the zebrafish. Here you can see the video, which is slowed down about a factor of 2. We quantified the fluorescence intensity at different regions, at the heart chamber, the inlet, and then outlet, and also a reference region somewhere else. And you can clearly see the frequency of the heart, the pulsing in the heart. Of course, the inlet and the outlet are in different phases, 180-degree phase shift. And which we can also quantify here. Interestingly, because this is a color camera, we were exciting with blue light. So we still get the green and the red channel. And we noticed a phase delay between the red and the green channel and the pulsing of the heart. And looking at the literature search, other people have already found that. And this was from the, in their case, they were labeling the surrounding tissue and the red blood cells with 2 different proteins. And they found a phase shift between the contraction of the tissue and the actual blood cells moving. In our case, we were just dealing with autofluorescence, which seems to be somewhat specific to these different kinds of cells and tissue types.